Maureen Richards, and this video is to go over the general characteristics of a really important group of bacteria, the Streptococci. The Streptococci and the Enterococci, which we include in some of the classifications for the Streptococci, are a really diverse group of gram-positive cocci. They contain more than 100 species of organism and cover a bewildering variety of different diseases. And that can make them a little bit confusing. There are actually 3 overlapping schemes that are used to classify the different species of Streptococci and Enterococci. This is a really important group to know. STEP loves them, and part of the reason for this is anyone can get infected at any time. It affects the very young and the very old, and the consequences of infections from streptococcal organisms can be none, where you just clear it and you're fine, or it can be normal flora, all the way to chronic disease or even life-threatening. So first off, how do we classify them? So in general, the Streptococci are gram-positive organisms. You can tell this by, on a gram stain, their typical royal purple hue, which you can see here. They tend to form pairs, as you can see down here, or chains, as you can see here. On blood plate agar, they show small, colorless colonies and sometimes can have different types of hemolysis, or how much red blood cells that they're capable of lysing. They can grow in a wide variety of environments. They can be either completely aerobic, or they can also be facultatively anaerobic, where the presence of oxygen they need, or it doesn't really bother them, but they're capable of respiration or fermentation. One of the main diagnostic features that it's worth keeping an eye on when thinking about Streptococci are that they are catalase negative. Why is this important? Well, the reason for this is that Staphylococci, which are also gram-positive cocci, are catalase positive. So if you have a gram-positive cocci organism that is catalase negative, chances are it's a Streptococci. If it's catalase positive, chances are it might be a Staphylococci. So that can be a really important biochemical test for differentiating them from a different organism. So, as I mentioned before, there are basically 3 overlapping schema that are utilized to classify the different groups of Streptococci. They are the Lancefield groups, their hemolytic pattern on blood plate agar, and then a wide variety of biochemical tests that basically describe how they grow and what enzymes they contain. So let's start with the Lancefield groups. The Lancefield groups were defined in the 1930s by a microbiologist by the name of Rebecca Lancefield. She found that the beta hemolytic strains of the Streptococci possessed group-specific cell wall antigens, most of which were carbohydrates. And we make IgM antibodies to these group-specific carbohydrates. So when you have a known group antibody that binds to a specific strep strain and agglutinates to the bottom, as it's doing in this well here, you can then know its Lancefield group. It's important when you're studying for STEP to know both the Lancefield group and the species name of the organism. For instance, Group B Streptococcus, which is an important streptococcal organism that we think about when we think about babies and meningitis, is actually Strep agalactiae, whereas the organism that causes strep throat most of the time is known as Strep pyogenes, but it's also known as Group A strep. And you might see it written both ways, which is why it's important to know both the Lancefield grouping and the Latin name of an organism. All right, the next way that we tend to classify strep is by their hemolytic pattern. Literally, when you grow it on blood plate agar, what does it look like? And there are basically 3 ways that hemolysis is characterized. There's beta hemolysis, which is complete. And if you look in, let me switch my color. If you look over here on this blood plate agar, this is an example of beta hemolysis. You can see through the B here. So you can actually see through it where the red blood cells have been cleared. There's also alpha hemolysis, which you can see down here. Alpha hemolysis is incomplete. And actually what it looks like is almost like a darkening. And if you hold the plate at like a 45-degree angle and look at it, it'll actually have a little bit of a green tinge. So it's no accident that I chose a green color here. Gamma hemolysis is literally not hemolytic. So no hemolysis, you can't see through it. It's not changing the color of the agar. It's growing on the agar. So you've got beta hemolysis, which is complete, alpha hemolysis, which is incomplete, and gamma hemolysis, which is no hemolysis. The last method we use is a wide variety of biochemical tests, which I'll discuss in later videos as I discuss each organism individually. And these can include things like its ability to survive in bile, its sensitivity to various antibiotics like bacitracin, or a metal-coated disc, a copper-coated disc, optochin, or its ability to secrete enzymes, as is the case with the CAMP test. So now that we know the different schemes that are used to classify our various groups of streptococci, let's look at kind of how they all fall out. So I've already mentioned that the organism that we most often associate with strep throat is S. pyogenes, or Streptococcus pyogenes. This is group A strep, and it has a beta hemolytic pattern. The most important biochemical test for it is whether or not it's sensitive to bacitracin. So literally, to find this out, you would have a blood plate agar and you put a little disc of bacitracin on it, and you would see that you would have a zone of inhibition where the organism was not growing on that plate. The next group is Lancefield group B, and as I mentioned before, that's S. agalactiae. It also has a beta hemolytic pattern, but in this case, it's bacitracin resistant. And it also has this factor known as CAMP, which leads to an enhanced zone of beta hemolysis when it's plated alongside Staph aureus. So this is one way that you could tell it apart from S. pyogenes, because S. pyogenes doesn't have CAMP factor, and S. agalactiae will not show a zone of inhibition around a bacitracin disc. There are some streptococcal organisms that do not have those Lancefield carbohydrates sticking out. So if the carbohydrates are hidden, then we can't make antibodies to them, in which case they are not groupable. Specifically, Streptococcus pneumoniae, which is a very important but not the most common cause of pneumonia, is not groupable. It's also a gram-positive cocci, and it has an alpha hemolytic appearance on blood plate agars. So you can see it, but it's got that kind of green tinge. You can tell this one apart from some of the other ones because it's bile soluble. Basically, if you put it in a test tube full of bile, all the bacteria will lyse. It's also sensitive to optochin, which, as I mentioned before, is kind of a copper salt-coated disc that if you put it on blood plate agar, once again, you're going to see that zone of inhibition. There's a group of streptococci known as the viridan streptococci. They're also not groupable. Here are two of the more notable viridan streptococci, S. mutans and S. sanguinis. Other examples would be S. bovis and S. mitis. These are also alpha hemolytic. The way you could tell them apart from S. pneumoniae is that they are not bile soluble. They'll actually survive in bile, and they're resistant to optochins. These are pretty hardy organisms. And then I have a group down here that is evidently not a streptococcus. It's actually an enterococcus. Well, that's kind of not entirely true. They used to be streptococci, but then they later got their very own group. They were all grown up, and now they're enterococci. But I include them on this table because they respond to a Lancefield grouping, and they are the group D organisms. And they used to be known as streptococci, so I put them on the table just to kind of keep everything neat and tidy. But your group D organisms that are your enterococci, they are gamma hemolytic. In other words, they are not hemolytic. They're also bile resistance, which you should have been able to figure out because of the kind of suffix of this word. Anytime you see entero, that means it's probably doing something in your gastrointestinal tract, which means it's probably got to be pretty hardy and can survive against bile. So as I mentioned before, I'll go through all of those tests in detail as I go through the various organisms, but it's a good idea to keep track of those tests because often a step stem will read something like, your patient is a 1-week-old infant presenting with signs of nuchal rigidity and fever. A spinal tap was performed, which showed gram-positive cocci that are bacitracin resistant and CAMP positive. And then you should be able to go, oh, that's group B strep. And then maybe the answer choice will say S. agalactiae. So that's kind of why all of this information is in this video, so that from these kind of complex vignettes, you can put them all together. And one way to do that is to just remember your ABCs. So if you use the Lancefield grouping, the test used to define a Lancefield group is always one alphabetic letter off from the actual Lancefield group. So group A strep, you're going to use bacitracin. Group B strep, you're going to use CAMP factor. Group D, Enterococcus? Well, yeah, it works, because you're going to use escalin agar, which basically shows that it's bioresistant. And then for Streptococcus pneumoniae, so pneumococcal strep, you're going to use optochin resistance. Those are kind of the basics of the Streptococcus characteristics and the tests that are used to identify them. So now go ahead and watch the videos on the Streptococci. Specifically for your first case, you're going to want to watch the video on group A strep.